Welcome to the fascinating world of neuroscience. Today, we are going to explore the molecular basis of learning, uncovering exactly how your brain changes its physical structure every time you learn something new. Whether you are memorizing a mathematical formula or learning to play the piano, your brain is constantly rewiring itself. We will build our understanding from the ground up, starting with the biology of brain cells and moving all the way to the mathematical equations that model human memory. To understand this microscopic process, let us first imagine walking through a dense, overgrown forest for the very first time. You have to push heavy branches out of the way, and your progress is incredibly slow and difficult. However, if you walk that exact same path every single day, the trail eventually becomes wide, clear, and effortless to travel. Your brain works in the exact same way when you are learning something new. The connections between your brain cells, which are called synapses, become stronger and more efficient the more they are used together. This dynamic ability to physically change wiring based on experience is the foundation of all learning. Now that we understand the forest analogy, let us look at the biological term for this process. At the most fundamental biological level, learning is driven by a process known as synaptic plasticity. Synaptic plasticity is simply the ability of synapses to strengthen or weaken over time in response to increases or decreases in their activity. The most famous mechanism for this strengthening is called long-term potentiation, often abbreviated as LTP. During long-term potentiation, when a sending neuron repeatedly and persistently stimulates a receiving neuron, the physical connection between them is permanently enhanced, cementing the memory into your brain. So, how does this strengthening actually happen at the cellular level? This enhancement happens because of special molecular gates on the receiving cell, specifically the NMDA and AMPA receptors. When the neurons fire together repeatedly, these receptor gates open up and allow calcium ions to flood into the receiving cell. This sudden calcium influx is the critical trigger. It starts a cascade of chemical signals that ultimately build more receptors and physically enlarge the synapse. Just like widening the forest path, the brain is physically building a wider bridge for signals to cross, making future communication much faster. Understanding these microscopic gates is not just biology, it is the essence of who we are. The molecular basis of learning explains human memory, identity, and skill acquisition. Without synaptic plasticity, you would be completely unable to form new memories. This tragic condition is known as anterograde amnesia. It was famously observed in the historic patient HM, who lost his ability to form new memories after the brain regions responsible for long-term potentiation were surgically removed. Every single thing that makes you who you are is stored in the microscopic, shifting weights of these neural connections. Beyond forming new memories, this plasticity is crucial for holding on to the ones we already have. Understanding these exact molecular mechanisms allows scientists and doctors to develop treatments for severe neurodegenerative diseases, such as Alzheimer's disease. In these conditions, the precious synaptic connections that hold a person's memories and identity are slowly lost over time. By studying how synapses strengthen and weaken, researchers hope to find ways to halt this decay and protect the brain's architecture. The health of these microscopic connections directly dictates the health of the human mind. Now that we have explored the biology, let us translate this into mathematics. To truly understand how learning works, computational biologists use mathematical models. The most famous is Hebb's rule, named after Donald Hebb, who famously said that neurons that fire together wire together. In this equation, delta W represents the change in the synaptic weight, which is the physical strength of the connection. The symbol eta represents the learning rate, determining how quickly the brain adapts. The variable x is the firing rate of the sending neuron, and y is the activity of the receiving neuron. If either neuron increases its firing rate, the synaptic weight grows much faster, perfectly modeling how repeated practice builds stronger memories. Let us see this formula in action with a concrete example, like learning a new guitar chord. We want to calculate how much the synapse between your motor cortex and your finger muscles strengthens. We are given an initial synaptic weight of 0.5 and a learning rate of 0.1. The sending neuron fires at 10 signals per second, and the receiving neuron fires at 20 signals per second. We substitute these values into Hebb's equation, multiplying 0.1 by 10 and then by 20. This gives a change in weight of exactly 20. Adding this to our initial weight, the new synaptic weight is 20.5. This massive increase represents a powerful surge in synaptic strength due to intense practice. Our calculation showed a massive increase in synaptic strength, which brings us to a major biological problem. The basic Hebbian rule has a critical flaw. If the neurons keep firing, the synaptic weight will grow infinitely large. In a real biological brain, infinite growth is physically impossible. A synapse cannot grow forever. It has physical and metabolic limits. If synapses grew without bounds, the brain would become unstable and unhealthy. Therefore, we need a mathematical way to represent the natural limits of biology, ensuring that our models reflect the true, stable nature of human learning. To fix this issue of infinite growth, scientists use a modified equation called Auger's rule. Auger's rule introduces a mathematical forgetting or stabilization term that prevents the synapse from exploding with infinite strength. The symbols delta W, eta, x, and y mean the exact same things as before. The crucial addition is the term y times w inside the parentheses. This acts as a biological penalty or decay factor. As the synaptic weight gets larger, this penalty term also grows, which eventually slows down the learning process. This formula beautifully states that a synapse will strengthen when neurons fire together, but only up to a certain natural limit, keeping the brain healthy and stable. Let us apply this new rule to the exact same neurons to see how it prevents runaway growth. We use the same initial weight of 0.5, sending rate of 10, and receiving rate of 20. We will use a smaller learning rate of 0.01 to observe the stabilization. We carefully substitute all our known variables into Auger's formula. Following the order of operations, we evaluate the terms inside the parentheses first. Multiplying the receiving rate of 20 by the current weight of 0.5 gives us 10. Subtracting this from the sending rate of 10 leaves us with 0. Multiplying the outside terms by 0 means the entire change in weight is 0. Physically, this beautiful result means the synapse has reached its maximum optimal strength and stabilized. Now that we have calculated the limits of synaptic growth, let us visualize the actual biological structure we have been modeling. On the screen, you are looking at a schematic diagram of a biological synapse. On the left is the presynaptic input, representing the variable X, filled with neurotransmitters ready to be released. The arrow crossing the gap represents the synaptic weight, or W, which is the strength of the connection we just calculated. On the right is the postsynaptic output, representing the variable Y, featuring the AMPA and NMDA receptors we discussed earlier. This diagram perfectly captures the physical intersection of biology and mathematics, showing exactly where Hebbian learning takes place. We have covered a lot of ground today, so let us summarize the most important ideas. First, learning is driven by synaptic plasticity, specifically long-term potentiation, where brain cells physically alter their connections based on experience. Second, we can quantify this biological magic using Hebb's rule, which proves that neurons that fire together wire together. Finally, Auger's rule adds a necessary decay factor to ensure these connections maintain healthy, stable limits. You have now mastered the molecular and mathematical basis of how your own brain learns.